All living organisms contain cells which require nutrients and oxygen to produce energy. Cells also produce wastes such as urea, uric acid, and carbon dioxide. Hence, there should be a continuous supply of nutrients and removal of waste from the cells. This process of supply and removal is carried out by simple organisms like sponges by allowing water to flow through their body cavities. It is also carried out by complex organisms like humans through a special medium called body fluids that circulate these substances throughout the body. Body fluids include blood, interstitial fluid, and lymph. Blood is the common body fluid used for circulating nutrients and removing waste throughout the body in humans. It is a liquid connective tissue that consists of 55% liquid plasma and 45% formed elements called blood cells. Now, let's see what plasma is made of. Plasma is a straw-colored fluid constituting 90 to 92% water, 6 to 8% proteins, 1.2% nutrients and gases, and 0.8% inorganic salts. The major proteins in plasma include albumins, globulins, and fibrinogens. Each type of protein performs a specific function. For example, albumins maintain the osmotic balance by transporting minerals in and out of the blood. Globulins help in the body's defense mechanism, while fibrinogens are crucial for the clotting of blood. Plasma also contains organic nutrients such as glucose, amino acids, and fats, respiratory gases such as oxygen and carbon dioxide, and inactive clotting factors. There are also traces of inorganic salts such as sodium, calcium, magnesium, and bicarbonate in plasma. The remaining 45% of blood is made up of formed elements that include erythrocytes, leukocytes, and platelets. Erythrocytes, or red blood cells, comprise 99% of formed elements. In adults, they are formed in the bone marrow. Did you know that a cubic millimeter of blood contains around 5 to 5.5 million RBCs? RBCs are biconcave in shape and lack a nucleus in the mature state. Their red color is due to the presence of an iron-containing protein called hemoglobin, which plays a vital role in the transport of the respiratory gases, oxygen and carbon dioxide in the blood. The lifespan of an RBC is around 120 days, after which it is destroyed in the spleen, which is known as the graveyard of RBCs. A low RBC count in the blood can cause anemia in humans. Unlike erythrocytes, leukocytes or white blood cells are colorless and possess a nucleus. There are 5 types of leukocytes. Based on the presence or absence of granules in their cytoplasm, these cells are further grouped as granulocytes and agranulocytes respectively. Neutrophils, eosinophils, and basophils are called granulocytes, while lymphocytes and monocytes are called agranulocytes. The chart displays the composition of different types of WBCs in blood. Each of these cells has a specific function in the defense mechanism of the human body. For example, neutrophils and monocytes act as phagocytic cells. and destroy foreign substances by engulfing them. On the other hand, eosinophils fight infections and are linked to allergic reactions, while basophils, the smallest granulocytes, secrete substances such as histamine and serotonin and are involved in inflammatory reactions. In contrast to the other leukocytes, lymphocytes include T lymphocytes and B lymphocytes and are involved in the immune responses of our body. For example, T lymphocytes attack abnormal or tumor cells, while B lymphocytes produce antibodies against foreign microorganisms. Did you know there are around 4,000 to 11,000 WBCs per cubic millimeter of blood? The third component of formed elements is platelets, which are formed in the bone marrow as tiny cell fragments called thrombocytes. A cubic millimeter of blood contains around 150,000 to 450,000 platelets. They circulate in the blood for around 7 to 10 days, after which they are destroyed in the spleen and liver. Platelets help in clotting by releasing calcium and clotting factors present in their cytoplasmic granules. Let us see how platelets help the clotting of blood. Clotting or coagulation is a protective mechanism that takes place at an injured site to prevent loss of blood. An injured blood vessel releases certain chemicals which trigger the activation and accumulation of platelets at the injured site. The activated platelets in turn release other factors required for clotting. These factors, along with thrombokinase and calcium, activate the inactive clotting components of plasma to start a cascade of reactions. These reactions catalyze the conversion of inactive prothrombin to active thrombin, which in turn helps convert inactive soluble fibrinogen into active insoluble fibrin. Fibrins form a mesh in which dead and damaged blood cells are trapped to stop blood loss. The mesh looks reddish-brown and is called a clot or coagulum, which is formed in 3 to 5 minutes. As platelets help clotting, their reduction can result in clotting disorders causing severe blood loss. Apart from blood, interstitial fluids and lymph are other important body fluids that make up our body. Interstitial fluid or tissue fluid is formed when some quantity of water and water-soluble substances squeeze out from the blood capillaries into the intercellular spaces. Its composition is similar to that of plasma and serves as a medium between blood and cells in the exchange of nutrients, gases, and waste. When this tissue fluid enters the lymphatic capillaries, it is called lymph. Lymph is a colorless body fluid that contains lymphocytes, nutrients, hormones, and proteins. Lymphocytes in the lymph help the body fight microbes such as viruses and bacteria. Lymph also plays a major role in the absorption of fat through lacteoles in the intestinal villi, where the absorbed fat is drained into lymphatic vessels. This network of lymphatic vessels that collects lymph fluid forms the lymphatic system. The collected lymph fluid, along with absorbed fats, is drained into the blood at lymph nodes. In this manner, lymph, blood, and interstitial fluid play an important role in the continuous supply of nutrients to cells and the removal of waste from cells. Do you know what your blood group is? Blood in humans can be grouped based on the presence or absence of special proteins called antigens on the surface of the red blood cells or RBCs. Based on these differences, Karl Landsteiner categorized blood groups into the ABO and Rh blood groups. The ABO blood groups are differentiated based on the presence or absence of antigens A and B on the RBCs, while the Rh blood groups are differentiated based on the presence or absence of the Rh antigen on the RBCs. The different ABO blood groups are referred to as blood group A, B, AB, and O. The presence of the Rh antigen in the blood is called Rh positive, and the absence of the same is called Rh negative. Did you know that the Rh antigen is named after the rhesus monkey in which it was first identified? Apart from antigens, anti-A and anti-B antibodies are naturally present in the plasma of humans. However, the presence or absence of a particular type of antibody depends on the antigens on the RBCs. For example, individuals with A blood group have A antigen, and hence the anti-B antibody is naturally present in them. Similarly, individuals with B blood group have B antigen, and hence the anti-A antibody is naturally present in them. On the other hand, individuals with AB blood group have both A and B antigens, and hence antibodies are absent in them. In the case of individuals with O blood group, antigens are absent, and hence both anti-A and anti-B antibodies are naturally present in them. These details are critical during blood transfusion between individuals, because specific antigens and antibodies present in the blood react and cause the clumping or agglutination of RBCs. Hence, blood group matching is carried out before blood transfusion. Blood group matching is a means to check if donor and recipient blood match or are compatible with each other. First, a small quantity of blood is collected from the recipient. Then the blood is allowed to clot, which produces an extract, that is, an amber-colored fluid called serum. Finally, the recipient's serum is added to the donor blood to check for agglutination. If agglutination occurs, both blood groups are said to be incompatible, and so blood cannot be transfused. However, if agglutination does not occur, both blood groups are said to be compatible and transfusion can be carried out. This table showcases the results of ABO blood group matching between different blood groups of donors and recipients. From these results, it is clear that individuals with blood group O can donate blood to all blood group types, but receive blood only from individuals with blood group O, whereas individuals with blood group AB can receive blood from all blood group types, but donate blood only to individuals with blood group AB. Hence, individuals with blood group O type are called universal donors, while individuals with blood group AB type are called universal recipients. In addition to ABO blood grouping, Rh blood group matching is also universally carried out during blood transfusion. This is because if Rh positive blood is transfused to an Rh negative person, it can induce the formation of Rh antibodies, resulting in the agglutination of RBCs. An example of such incompatibility takes place in an Rh negative woman carrying an Rh positive fetus. In such cases, the Rh incompatibility results in a condition called erythroblastosis fetalis. In most cases, the first child will not get affected because the blood of the mother and child are well separated by the placenta. However, during the delivery of the first child, a little of the Rh positive blood from the child can pass through the placenta and mix with the mother's blood. This induces the formation of Rh antibodies in the mother's blood. The problem occurs during the second pregnancy, when the Rh antibodies present in mother's blood cross the placenta and destruct the fetus's RBCs. This results in severe anemia and jaundice, and at times fatality of the fetus. Erythroblastosis fetalis can be prevented by injecting anti-Rh antibodies, commercially called RhoGAM, into the Rh negative mother immediately after her first delivery. Thus, Rh and ABO incompatibility can be easily prevented by knowing the blood group types. The circulatory system or cardiovascular system refers to the organs and blood vessels involved in the transport of blood throughout the body. It is of two types, open and closed. An open circulatory system is found in arthropods and mollusks. In these organisms, the blood pumped from the heart reaches the open spaces or body cavities called sinuses through large blood vessels and bathe all the organs. Whereas a closed circulatory system is found in annelids and chordata. In these organisms, the blood pumped from the heart passes through the closed blood vessels to reach the tissues. The heart, the vital organ of blood circulation, is made of cardiac muscles. It has many chambers which vary between animals. For example, fishes have a two-chambered heart comprising one atrium and one ventricle, while all amphibians and reptiles, with the exception of crocodiles, have a three-chambered heart comprising two atria and one ventricle. On the other hand, all crocodiles, birds and mammals have a four-chambered heart comprising two atria and two ventricles. The blood circulation in these animals also differs due to the difference in the number of chambers. For example, fishes exhibit single circulation. Deoxygenated blood is pumped from the heart to the gills for oxygenation, and the oxygenated blood directly flows into the tissues, whereas deoxygenated blood collected from the tissues is returned to the heart. In this way, blood passes through the heart only once during circulation, and hence this is called single circulation. Frogs and reptiles exhibit incomplete double circulation. Oxygenated blood from the lungs or gills or skin reaches the left atrium, while deoxygenated blood from body tissues reaches the right atrium. Blood from the left and right atrium is mixed in the single ventricle. Thus, the blood passes through the heart twice and gets mixed in the single ventricle during circulation, and hence this is called incomplete double circulation. Birds and mammals exhibit double circulation. Oxygenated blood from the lungs reaches the left atrium, while deoxygenated blood from body tissues reaches the right atrium. The blood from both the atria is released into their respective ventricles, thus preventing any mixing. In this way, blood passes through the heart twice during circulation, and hence this is called double circulation. During blood circulation, the heart pumps blood to all the tissues through a set of blood vessels called arteries, while the blood from the tissues reaches the heart through another set of blood vessels called veins. Arteries are large blood vessels that carry oxygenated blood from the heart and branch out to form small arterioles that end in capillaries to reach the tissues. These capillaries collect deoxygenated blood from tissues and converge to form venules, which finally converge to form the large blood vessels, the veins. However, there are two exceptions in which the artery and vein perform different functions. The pulmonary artery carries deoxygenated blood from the heart to the lungs, while the pulmonary vein carries oxygenated blood from the lungs to the heart. Now let's look at the structure of the human heart in detail. The heart is about the size of a fist and lies in the thorax between the two lungs, slightly tilted towards the left lung. It is protected by a double-walled membranous sac called the pericardium, which encloses pericardial fluid. It has 4 chambers that include 2 upper small atria and 2 lower large ventricles. A thin muscular wall called the interatrial septum separates the 2 atria, while a thick interventricular septum separates the 2 ventricles. Besides, a thick fibrous tissue called the atrioventricular septum separates the atria from their respective ventricles. However, there are openings between the chambers of the heart which are guarded by 3 types of valves that allow the flow of blood in one direction. The tricuspid valve is made of 3 muscular flaps or cusps and guards the opening to the right atrioventricular septum. The bicuspid or mitral valve is made of 2 muscular flaps or cusps and guards the opening to the left atrioventricular septum. The semilunar valves guard the opening between the right ventricle and pulmonary artery and between the left ventricle and aorta. When these valves close, they produce a lub-dub sound heard during a heartbeat, while the closure of the tricuspid and bicuspid valves produces the first sound lub. The closure of the semilunar valves produces the second sound dub. Also, specific regions in the heart consist of specialized cardiac muscles called nodal tissues. The first nodal tissue, the sinoatrial node or SAN, is present in the top right corner of the right atrium. On the other hand, the second nodal tissue, the atrioventricular node or AVN, is present in the bottom left corner of the right atrium near the atrioventricular septum. Another group of nodal tissues starts as an atrioventricular bundle or AV bundle from the AVN and passes through the atrioventricular septum and then through the interventricular septum. This finally branches into right and left bundles in the ventricles to form minute Purkinje fibers over the ventricles. All these fibers together form the bundle of His. Here, the SAN can automatically generate around 70 to 75 action potentials or electrical impulses per minute. This action potential is transmitted to every other part of the heart through the AV node and bundle of His to bring about the rhythmic contraction of the heart. This is why the average heart beats 72 times per minute. Hence, the SAN is called the pacemaker of the heart. In some cases, An artificial pacemaker is used in individuals suffering from heart problems relating to the SAM, which generates action potentials or electrical impulses in place of the natural pacemaker, the SAM. Thus, the transmission of action potential to different parts of the heart is essential for blood circulation. The heart is the only organ in humans responsible for the circulation of blood. It contains 4 chambers: the right and left atria and the right and left ventricles. As we know, double circulation takes place in organisms with a 4-chambered heart. Double circulation involves pulmonary and systemic circulation. Pulmonary circulation refers to the circulation of blood between the heart and the lungs, whereas systemic circulation refers to the circulation of blood between the heart and the tissues of the body from head to toe. Thus, pulmonary circulation oxygenates blood in the lungs and returns it to the heart, while systemic circulation supplies oxygenated blood along with nutrients from the heart to all the tissues and returns wastes along with deoxygenated blood from the tissues to the heart. Nutrients are added to the blood through the hepatic portal system, which connects the intestine and liver to the systemic circulation. Similarly, blood circulation to the heart is carried out by the coronary arteries and cardiac veins, which together comprise coronary circulation. When the heart pumps blood, a heartbeat is generated by the systole or contraction and diastole or relaxation of the chambers of the heart. A heartbeat involves a sequence of events that repeats, forming a cardiac cycle. On average, our heart beats 72 beats per minute, which means 72 cardiac cycles per minute. Thus, one cardiac cycle takes 0.8 seconds. The cardiac cycle starts with the 4 chambers in a relaxed state or joint diastole. Atrial pressure causes the bicuspid and tricuspid valves to open. This allows blood to flow from the vena cava and pulmonary vein into the right and left ventricles through the right and left atria respectively. The semilunar valves are closed at this point. This is followed with the SAN producing an action potential that induces both the atria to contract at the same time, forcing more blood into the ventricles. Ventricular pressure increases due to the increased volume of blood, which forces the closure of the tricuspid and bicuspid valves and thereby prevents a backflow of blood into the atria. The action potential from the SAN then travels down through the AVN and passes onto the ventricles through the bundle of His. This results in a ventricular contraction or systole as well as atrial relaxation or diastole. The ventricular systole also causes the opening of the semilunar valves of the pulmonary artery and aorta. This causes blood to pump from the right and left ventricles to the pulmonary artery and aorta respectively. This is followed by a ventricular relaxation or diastole, causing the closure of the semilunar valves to prevent any backflow of blood from the pulmonary artery and aorta to the right and left ventricles respectively. At the same time, atrial pressure causes the tricuspid and bicuspid valves to open to let the blood flow from the atria to the ventricles. Now the four chambers are once again in a relaxed state or joint diastole. This sequence of events is repeated again as the SAN generates another action potential, resulting in the cardiac cycle. Cardiac output or efficiency of the heart is calculated using the volume of blood pumped by each ventricle per minute. Normally, each ventricle can pump around 70 milliliters of blood per minute, which is also called the stroke volume. So, cardiac output equals the product of the stroke volume and the number of beats per minute. In a healthy individual, the cardiac output is 5 liters per minute. But our body has the ability to increase cardiac output by 4 to 5 times during exercise and decrease it to return to normal at rest. Thus, the body can vary the cardiac output by controlling the cardiac cycle, which is driven by the transmission of action potentials. The cardiac cycle involves systole and diastole and is carried out automatically through nodal tissues of the heart. Hence, the heart is also termed myogenic, which means originating in the muscles. Our body has the ability to increase or decrease cardiac output when required. This is because nodal tissues, which induce cardiac activity, are controlled by the medulla oblongata of the brain through sympathetic and parasympathetic nerves of the autonomous nervous system or the ANS. While sympathetic nerves increase cardiac output, parasympathetic nerves act antagonistically to decrease cardiac output and bring it back to normal. Did you know that cardiac output can be measured using an instrument? The instrument used is called an electrocardiograph, which records the electrical activity of the heart as voltage traces. This recording is called an electrocardiogram or ECG. It provides information on the rate of heartbeat and the size and position of the chambers of the heart. An ECG is taken by attaching electrodes from the instrument to the patient at the wrists, ankles, and on the chest. An ECG captures the electrical activity of the heart due to atrial and ventricular contractions as voltage traces, which are represented by the letter P to T. A standard ECG should resemble this. Here, the P wave corresponds to the contraction or depolarization of the atria, that is, the atrial systole. It is followed by the QRS wave, which corresponds to the contraction or depolarization of the ventricles, that is, the ventricular systole. The final T wave corresponds to the relaxation of the ventricles to the normal state or repolarization, and the end of the systole. The number of QRS complexes equals the number of heartbeats. Hence, any deviation from the standard ECG signals a problem relating to the heart. Most heart-related problems are related to drastic changes in blood pressure. Now let us see what blood pressure is. During circulation, when the heart pumps, blood rushes through the blood vessels, exerting some pressure on the walls of the blood vessels. This is called blood pressure or BP. The heart undergoes alternate systole and diastole, and hence blood pressure is measured as systolic or pumping pressure and diastolic or resting pressure. Did you know that blood pressure is measured using an instrument called a sphygmomanometer? It measures blood pressure in millimeters of mercury or mmHg. A healthy individual has a systolic and diastolic pressure of 120/80 mmHg. When BP is 100/50 or less, it is called low blood pressure or hypotension. Similarly, when BP is 140/90 or more, it is called high blood pressure or hypertension. Thus, any drastic change from normal blood pressure leads to coronary artery disease. Coronary artery disease or CAD affects the coronary circulation of the heart. Calcium, fats and cholesterol together accumulate and form plaque in the coronary artery and reduce or obstruct the blood supply to the heart. This accumulation of plaque is called atherosclerosis. Some of the symptoms of CAD include angina, heart attack, cardiac arrest, or heart failure. Angina or angina pectoris is severe chest pain due to a reduction in the supply of blood to an area of a heart muscle. Whereas a heart attack results when there is a total blockage of blood supply to the heart muscles, causing the patient's death. On the other hand, cardiac arrest is the sudden stopping of the pumping of the heart due to failure of the contraction of the heart. In contrast to cardiac arrest, heart failure or HF is caused due to the inability of the heart to pump enough blood to meet the body's demands. This can result in the accumulation of body fluids, often leading to swelling of the legs and ankles, and also congestion of the lungs. Hence it is called congestive heart failure. Thus, cardiac activity is well regulated, and any deviation from normal activity may lead to heart diseases.